Hello sir, how are you?
I'm fine.
So we are starting with the first question of the podcast.
Yeah.
When bacteria is growing in an environment,
Mm-hmm.
what affects more? Is it nutrients or other factors to its growth?
Okay. So, like you mean that if a particular bacterium is growing in its natural environment, or when we are growing the bacterium in the laboratory, what should be our prime target to consider? Like, if I want a maximum growth of the organism, I should focus more on the nutrient requirements of the organism, or I should focus on other physical requirements like the temperature, pH. Is it that what you are
Yeah.
trying to ask? Okay. So basically, you see that it's a complex interplay. You just cannot depend on only one particular factor alteration to alter the growth of a particular organism. At a time, we keep on changing one or the other parameters, and then we keep on analyzing that what is the effect of its change in the growth pattern of the organism or the growth rate of the organism. But then it is an interplay of growth. You must have heard of the Liebig's law, which is generally more applied to higher organisms. Like the total yield or biomass of a particular group of organism, it would be dependent on the nutrient concentration which is lowest, depending on the growth requirements of that particular organism. We know that cell on an average will require around a lot of water, because we know that 70 to 80% of it is water. Then talking about the major elements, like you have your carbon, hydrogen, oxygen, nitrogen, phosphorus, and your sulfur, the knops, what we call. So at a time, like if the organism is like starving for phosphorus, say for example, like if the phosphorus is near its limiting amount, the minimum amount which the organism requires in nature also, or in the medium also, the phosphorus which you have provided is very less, and you think that you want to increase the growth of the organism and you add a lot of nitrogen there. So obviously the organisms whose growth was halted because of nitrogen depletion, uh, that group of population will now start what you call as proliferating. So it is like which nutrient is required for the organism for its growth would be the one which is near its minimum value which is required for the growth of that particular organism. At the same time, you must have heard of the Shelford's law of tolerance. That actually says that it is not only the nutrients, when we consider the abiotic factors, it is not only the nutrients but all your other physical factors. It may be your temperature, it may be the redox potential, it may be your pH. So these actually set boundaries for the growth of the organism. You have the minimum and maximum. Probably when you were in your graduations and post graduation, we studied something called as optimum temperature or optimum pH, right? So every organism has a minima of its growth, and every organism will have its maximum of its growth. So according to this Shelford's law of tolerance, like you will find that if you go less than the minimum or higher than the maximum, that particular organism probably in the given period of time may be eliminated from that particular environment if that condition prevails for a longer period of time. So what I personally think, rather most microbial ecologists might be thinking, is that it is a complex interplay of these both. You need to focus both on nutrients as well as the physical factors. So it is both. So the answer is not one.
Okay. So change in one factor, does it affect the boundary of other factor? Like if we change the pH, does it affect the nutrient requirement by that microorganism?
Yeah, broadly speaking, it does. See, for example, if you are eating some food and you don't like that particular food, and then someone offers you like, if you eat this, I'll give you something good. Say a child does not have affinity towards milk, is not drinking milk. So you offer him, you finish that particular milk and I'll give you a chocolate or something like that. So presence of that chocolate in the near environment now forces that particular child probably to drink that milk, otherwise which he or she would have not actually loved to have. So similarly, what happens is if one of the factor is very conducive for the growth of the organism, like if there are like ample amount of nutrients, then probably the pH ranges may widen a bit, right? If say for example temperature is very conducive for the growth of the organism, then in that case again the pH ranges may be what you call as a bit widened. Like if the pH is more conducive, then the temperature ranges may widen. Because what happens is ki kuch to organism has one thing to like rely on, right? So with that favors the growth of the organism, and you may say that it empowers the organism to overcome the stress which would be created by the change in the other parameter. So that can happen, yes. Matlab not to a very great extent, but then yes, matlab if you are giving a proper temperature, then the pH ranges may like increase more. If you are giving a proper pH, then temperature ranges may increase more. If you are giving proper nutrients as required, right, not more or less or what, but then as required for the organism, then its capacity to overcome these other stresses, it automatically increases a bit.
So talking about stress, as you mentioned, the low availability of nutrients is also a stress. Like
Yeah.
starvation is also a stress.
Yes, yes.
So what strategies do microbes have to deal with starvation?
Yes. So starvation, matlab, see, it's like we need to really understand that we are talking about starvation. That means organism is living under a condition where it wants more food, right? And it is not getting that much amount of food. That time I'll say that it's starving, right? Uh, let us differentiate it from a group of organisms which we call as oligotrophs, which actually require a low nutrient concentration. So keeping that aside, talking about like when a cell is under starvation and cell thinks now it is not getting enough amount of nutrients, like what you will do, like you think about yourself, like if I am not getting enough food, if you are fasting, say for example, that is a kind of starvation. But if we are fasting, then what will happen is ki you see that you don't move out more, you stick to the bed more, and you see that your activities are to the minimum, right? So you want to save your energy, you want to live. So similarly the cell will do, like what the cell will do is it will reduce its metabolic activities. So all the metabolic activities of the cell, they are brought at a halt, so a reduced growth rate. Some general strategies what a cell will go for starvation is that it will reduce its growth rate, it will alter its metabolic pathways, it will alter its ways of substrate utilization, it may suspend its growth. You find like some cells, they go for production of structures like your endospores and such structures, like you have some references where spores have survived around 25-30 millions of years, some of the spores of bacillus, right? Then you have some changes in the surface structures which will what you call as put some porins, or it will add some receptors on the surface which will have a very high affinity for the substrates. So even if the concentration outside is less, they can work efficiently. So these you can say are some general strategies to overcome the stress of starvation. Like you reduce your growth rate, you suspend your growth, you have some surface receptors, you alter your metabolic pathways, you change your substrate requirements. So this is what generally a cell will do when it is starving.
Okay, so like you mentioned about fasting.
Huh.
When I am fasting, when people do starvation for a long period of time, they get skinny. So is there any morphological changes that bacteria have when it is starving?
As far as the morphology of the organism is concerned, matlab we are generally, like whenever you look under the microscope for a bacterial morphology, matlab for any undergraduate or postgraduate student, what you can see is a rod and a cocci, and that's it. Matlab, though there are many morphologies, but then this is what generally a graduation and post-graduation student like talks about morphology when I'm talking about. So as far as morphology is concerned, yes, skinny. I am imagining like a bacterium was a like full-fledged rod, and now it did not get it to eat, and now it has become a thin like flat cell. I don't know whether this really happens, but then you'll surely find that there are some changes at the what you call as the surface level, there are some changes at the metabolic level, there are some changes in the shape and the size of the organism, which may not be same for all. It may be different for different groups of organisms. Like if you talk about some cells of Vibrio, they will form what is called as the ultra microbacteria. So they are very small spheres which they'll form. They are not spores, but some very small cells with a very reduced what you call as requirement. So they will form that. Well, you will find that depending on the starvation of which nutrient is happening, like I told, like sometimes a cell may starve for carbon, sometimes a cell may starve for nitrogen. Like it is seen that if a typical E. coli cell is starved for carbon, it will start synthesizing 30 new different types of proteins. If it is starving for nitrogen, some 26 to 32 different types of proteins they are being synthesized. You will find like starvation for amino acids will like favor a stringent response. There would be a formation of what is called as a stringent factor, guanosine pentaphosphate and a guanosine tetraphosphate, a combination of that which acts as a stringent factor that would be synthesized. And this particular stringent factor, now it will see that any energy draining activity which is happening inside the cell, it is come to a halt. Like say for example, it will first go and stop the synthesis of rRNA. Now you know that this rRNA is a major component of the ribosomes. Ribosomes we know are the protein synthesizing factories. So amino acid is not there. So to synthesize basically a protein, what you are going to require is amino acids. So if there is no amino acid, this stringent factor will immediately go and like say to it, and transcription and translation will be completely halted. So first your transcription will be halted, so your rRNA is not synthesized, ribosomes are not formed, obviously your translation will be halted, so entire process of protein synthesis will come to a halt. Entire process of nucleotide synthesis will come to a halt. Peptidoglycan synthesis will come to a halt. So obviously the cell has signaled that you need to reduce your what you call as metabolic pathways. All energy draining activities, they will be reduced. Skinny again, I am again and again imagining like how I can correlate to it. But then morphologically, like no change. Like I don't know, like whether there would be a mini change in the morphology. As I told, like some cells they will reduce in size, that may happen. But then you will find that some proteins may come up on the surface. Like if a cell is starving, say for example for phosphate, so PHOE system, it will get activated, and that PHOE porin, it will go and what you call as come on the membrane, and that has a very high affinity for the phosphorus. So even the minuscule amount of phosphorus which is present, it will be immediately taken in by the cell. So this may happen. You will find like if phosphate is less, again if you look in the same scenario, you know that these gram positive organisms, they have structures on their cell which you call as teichoic acids, which have phosphate in it. Now phosphate is less, so cell cannot afford taking this phosphorus into teichoic acids. So they will start synthesizing teichoic acids devoid of phosphorus. This change you may see. So there will be formation of tecuronic acid, and that is now devoid of phosphate. Cell may go for, again talking about phosphate deficiency, I'll say. Cell may go for synthesizing enzyme, the PHO system which I told, that would be activated. And many different enzymes and proteins will now be formed. Cell up till now was relying only on the inorganic phosphate stuff which was present in the environment. Now the cell will start synthesizing enzymes like alkaline phosphatases, which will now what you call is also explore the organic sources of the phosphate. So these changes, it may happen. Like if there is a what you call as a deficiency of ammonia, right? The ammonia starvation, the NTR system would be activated. The more detail I go, the more complicated it would become. So I think this much would be fine to have an overview key. Depending on the which nutrient the cell is starving for, some changes you may see. You may find some cells, they are having some surface what you call as aggregators which are formed, which will help them to aggregate. This you will find in Vibrio species also. So such morphological changes, or you can say structural changes or structural modifications or metabolic modifications you will find when the cell is undergoing starvation, right? Because like you and me, they also want to live.
Yeah. So in my previous podcast with Dr. Amit Yadav on phytoplasma, he explained that phytoplasmas are unculturable organisms.
Right.
And the reason for that is that we are not able to figure out what is the nutrition requirement of those particular phytoplasmas are, and that is the reason why we are not able to synthesize synthetic media.
Great.
So why is that?
So true. See, it's like if there is some guest coming into your house, like you have invited someone to your house, probably for the first time you don't know what he or she likes to eat. If you are not providing proper nutrition to that particular organism or to that particular guest, if you are not offering the proper food which he or she likes, so then you conclude in a different way. Like say for example, if there is a particular guest who has come to your house and you serve all non-vegetarian food to him and he is a pure veg, he'll starve obviously. He'll not die, he'll go home and eat. But then in this case it is not like that. When I prepare a nutrient medium, what I do is that you talk to any person, generally we go and prepare a nutrient medium like which has 1 gram of peptone, which has zero point 0.5 grams of NaCl, which has 0.3 grams of yeast extract, which has 2.5 grams of agar, water, and with all those minerals. So probably we define a nutrient medium according to the maximum requirement. We thought that this will support the growth of the maximum organism. This is what we have been doing for many years. But then we are all aware of this great plate count anomaly. With these rising modern molecular biology methods to explore the microbial diversity, we now came to know that we have hardly explored only 0.1, somewhere 1, I don't think more than 1% of the bacterial diversity or microbial diversity at large. So whenever people come and ask me what scope microbiology has, I always say that there is 99% of the scope. Because whatever things are happening around and whatever products which we have come up with the microbes, that is only from the 1% of the diversity which we have explored up till now. So your question again, why is unculturability so common, and why organisms are still unculturable? We call them as yet uncultured. They are not unculturable. The fact that we have not been able to culture them in our laboratory does not mean that they are unculturable. So one of the reasons, to mention a few, there are many reasons you'll find out, just now we mentioned about starvation and oligotrophs. So oligotroph is a completely different class now. Oligotrophs are the organisms which will grow at a very low nutrient concentrations. Like for a copiotroph, which is growing at higher nutrient concentrations, you require additional nutrients at the concentration of around 50,000 milligrams per liter. Whereas these oligotrophs, they require hardly a concentration of around 50 to 100 milligrams per liter. So that is the huge difference. Probably these high nutrients are too toxic to them. You'll find they have very different morphology also. You'll find that some of them, they literally, some of the genera, they have radial symmetry. They literally look like starfishes. They have appendages. You must have heard of Colobacter or your Hyphomicrobium. And there are many other groups which look like starfish, which looks like sea urchin. So for them, having high nutrients is actually toxic to them. So probably we are not providing them the right amount of nutrients. See, more is not always right. It's not like a wedding function where you always say that the food should not be less, everyone should get maximum. So it's not like that. The high food content would be toxic to them. So that may be one of the reasons. Similarly, we never thought of growing them for a longer period of time. What we generally do is what you also might be doing, you incubate a plate and you don't have that much patience. We look it for after 24 hours.
Next day I want that colony.
Probably next day is a Sunday. Then you will get it on Monday.
Yeah.
And then maybe you forgot on that day, maybe the third day. So we don't go beyond 3 days. But there might be some organisms which are slow growers. In a class, you have some fast learners and you have some slow learners. You tell one thing to one person, he understands immediately. There are some who have already studied it from YouTube. Then there are some whom you tell 10 times, then they'll understand.
Yeah.
So if I am telling one thing to one person and he is understanding it, and the other person has not yet understood, that does not mean that he will never understand in his life. Probably it will take more time. So sometimes only time may be a factor. we think that every bacterial cell is going to multiply in 20 minutes. It's not like that. But so there may be slow growers. There may be certain organisms uh, which may be having a different pH requirement. See, we have decided everything that is normal according to probably what is normal for us. Like you ask to any person, you ask them like what is a normal pH. Like if I ask you what is a normal pH, what would you answer?
7.
7. Probably that is because the pH of our blood is 7.
Yeah.
I ask you what is a normal temperature?
37.
37. Why? Probably because see, most of the studies which we started with in microbiology, they focused on human pathogens. Therefore, it's not like we are not going to study organisms which are growing at pH 7. We are not going to explore the organisms which are growing at temperature 37. But you'll find that organisms are growing at 110 and 115.
Yeah.
Right? You'll find organisms are growing at minus 17. Right? You'll find probably organisms are not growing below 70. There are certain like hyperthermophiles, right? What we know. These are growing at temperatures not below 80. They'll never grow. So you think that at 37 it is not growing, it is not present in the environment. It's not like that, right? So these simple abiotic factors, what we discussed, as I told, you go below the minimum or above the minimum, they'll be eliminated. So you brought in a plate and you kept at 37. If it is below the minimum of certain, they will be eliminated. If it is above the minimum of certain, like certain psychrophiles will not grow at about 10. You are not going to incubate your plate in a refrigerator probably. But then you will find that temperature is there. Similarly, pH, as you told, like you don't adjust the pH of your medium to 3 or 9 or 11. You never thought of. But probably if you have got a sample from, say, hydrothermal vent, or you got a sample from acid mine drainage, probably that time you may think that I should alter the pH. You know?
Should mimic the
You
environment.
should mimic the environment, right? But then certain organisms which are thermophiles or certain organisms which are acidophiles, you will find very near to your environment where the conditions are not like acidic or thermophilic, but we miss them out.
Yeah.
So as you told, like this may be one of the strategies which we can do.
Yeah.
You know, so you can mimic the environment, you can simulate the environment. You know, so you can take these particular organism and try to find out like what exactly is present in the environment of the organism and how exactly we should grow it. So what is the temperature there, what is the pH there, and this is not something novel which we are doing now, right? Right, when these organisms were being discovered right from the times of Louis Pasteur, Robert Koch, they also, if you see, they added the calf serum, they added the soup from different what you call as broths and meats, and they were trying to grow these fastidious organisms which have a specific requirement. So you try to mimic the environment in the preliminary phase. Once you try to mimic the environment, grow them in your laboratory, then probably you will come to know that what exactly they require for their growth. Once you know that what exactly they require for their growth, then every time you will not mimic the environment, then you may add only that particular nutrient which is required for the growth of that particular organism. So, see, sometimes addition of more nutrients will increase the number of organisms on your plate, the diversity and the numbers. Sometimes reducing the nutrients, like I told, if you are like scavenging for your oligotrophs, so that time reducing the nutrients will increase the number of organisms and diversity on your petri dish. Or sometimes changing the other factors, as I told. Sometimes there are some organisms which are like ek duje ke liye. Like there is a synergistic association, like one organism is producing some chemical which is used as signal for the growth of the other organism. So we want to isolate them totally. That is also something like which will never happen, because one organism will not grow in the absence of the other.
Yeah.
So in that case, probably you will have to either identify that what chemical is being produced by this one organism for the growth of the other. Once you know that these two are growing together, now the next step would be to identify what this organism is doing for this. Then probably you will artificially provide that reagent or that chemical or that nutrient or that signal, and now probably you can separate it.
This similar